As you know from normal physiology, thyroxine in molecular doses speeds up metamorphosis, transition from the two-leg to the four-leg stage in frogs. And the research question, the main question was, what will happen when thyroxine in a homeopathic high dilution is applied? So an important thing for us was a multi-center approach. At that time was the University Graz even involved, and my close colleague Waltraud and Rolf van Weyck, who also I think is well known here, but he's at the University of Utrecht, so he's in both worlds, and others who did experiments in their centers or visited our center. And peer review process was always an important thing. So what we did had to be accepted by journals, and as you see, there are quite normal journals among them. I mean, homeopathy journals are a very important thing, including the International Journal of High Dilution Research, but it's also human veterinary toxicology or the FASIP journal or perfusion. And this is a picture of, you may recognize Esat Ernst. He's not an avid friend of homeopathy, but however, he invited at the time, he was, I don't know by what, he invited us to publish in one of the journals he edits, which is the Perfusion Journal, with the tadpoles. So this is a black dot on his biography that he invited us there to publish. And I have to say that we had a main line of research in this amphibian field, and we had side tracks. We had after the first years, we had to decide which are the side tracks, which are the promising tracks. So I, of course, present you the promising things here, but it has been a frustrating, at times a frustrating endeavor of research. I'd like to invite you to get one of the books out there. You can have them for free if you're interested. There is more about the frustration on the sidetrack. So if you like frustration, suffering, and so on, read the book. We had experience with lowland amphibians from normal countryside. Interesting results, but poorly reproducible, which I will not present you here. Experiments with highland amphibians with a high dilution, with interesting results and quite good reproducibility, which I will present you here. Highland amphibians mean they come from a highland biotope in the Alps. So they could be from Slovenia, but they come from the Austrian Alps. Just a little remark. I know we run out of time, but my wife and I love to walk in Slovenia. It's a really very nice now. Please. Well, they're Rana temporaria, green grass frog larves, which are meanwhile severely you cannot just take them out of the lakes today, so they are preserved by law there. So, and another thing is laboratory details. We try to stick to rigorous protocols, and when you work in multi-center, you have to write everything down you do, and you have to transfer this to the other colleagues that have to do the same thing you do. And this comes down to very, very minor details. You'll be astonished what could be done in other ways or wrong or different. So the target parameter is the frogs, the tadpoles shall reach the four-legged stage. What we use as a high dilution is thyroxine, so the hormone, in 30x compared to water analogously prepared. And of course, it was a double-blind experiment. I mean, the tadpoles were blind. We thought they were blind, but they realized what happened. And the researchers were blind, of course. And treatment started at the two-leg stage, and the test or control substance were added to the basin intervals. There were, as you see, quite extensive experiments at times with a good layout plan. And in total, I calculated more than 20 sub-experiments over the years. And well, this is a symbolic photo, I'd say. The group to the way around, to this side, would be the faster ones. They developed four legs. And this group would be the slower ones that developed they have only the hind legs and not yet developed the forelegs. So this is an idealization of what could happen. But it was visible that the water groups normally in the experiments, they developed faster than the thyroxine animals. So we have the timer running this direction and the number of four-legged animals this direction. And after the first measuring point, you see a difference between the groups, and this difference continues to even grows. So also with many animals, in many experiments, leads to the conclusion that the number of four-legged animals was lower in the homeopathically treated groups than in the control group. So metamorphosis would slow down, which is amazing for a biologist. For our zoologic colleagues, was also amazing. Why should it slow down? Homeopath said, yeah, of course it slows down because it's the opposite of the, according to the similarity principle, it's the opposite of normal thyroxine that speeds up. So homeopathy would slow down. Well, we believe that, it's okay. However, with other tricky experiments, when adding this substance at more frequent intervals, you can also speed the animals up, which is also within the frame of homeopathy as well, one of the features of homeopathy. You can have both reactions if you want. Here we focus on the slowing down. So yeah, these are the overall results of these 24 experiments done over 25 years. When the bar is going down, there is the effect of slowing down, yeah? So you see in most of the experiments, there was a slowing down from the first to almost the last. There were some exceptions, with 3 going the other way around, the other way. There were some with no results, no differences. It's also a result when you do not see a difference within groups. And well, the overall is that in the laboratory 1 in Graz, the difference was highly significant. In the second laboratory in Graz, highly significant. And the external researchers also reached highly significant overall result. So, and I want to refer to, you help me to pronounce that correctly. It's Juarez Ferreira, yeah? Okay, who made analogous frog and turret hormone experiments in Brazil. And I was very happy to learn that because they had really no connection to us. There was no knowledge of tricks or whatever. and they had very interesting and supporting results. Yeah, okay. And Leonie asked me to present, I think I have a couple of minutes more, experiments with thyroxine sealed in glass vials, because it matches so nicely to this conference. So this is, well, yeah, my laboratory colleague Waltraud. We'll see the reference for the lady. The information transfer, we don't know whether the information is transferred from the bottle to the lady's jacket or the other way around. And this is what we investigate. We put the thyroxine 30x in glass vials, sealed them, and so the water, and hung the vials into the aquaria with the basins, hoping that the vials would not leak, but exert some physics effect. Of course, they would not leak, yeah. And this is the same logic of presentation. You see the thyroxine group, the red one, is still below the blue one, the water control group. The difference is much smaller. On here you see the single experiments, most of them gone in one direction, so slowing down of metamorphosis. There were also two who did something else. This was also not only done by us, but by colleagues. And I must say that here we started to reflect on the fact that maybe the researcher plays a very crucial role in homeopathy research. So maybe there is some interaction between the researcher, his or her field or whatever, characteristics, and the homeopathy drug and the tadpole. Well, homeopathic doctors tell us, well, of course, there is an interaction between the therapist and the patient and the remedy. Yeah, of course. Well, this is embarrassing for laboratory research because the researcher strives not to be involved in her or his experiments, but then, yeah, there it is. We did a regression analysis over the decades of research, and it seems that between the start of these studies and now, highland biotopes became too warm to provide animals at the starting stage suitable for the experiments. So this is really a shame for homeopathy research, but also it's of course it's a sign of climate change. It's 1 degree. Well, anyway, so this regression analysis suggests, even if our laboratories are of course kept constant, but the pre-treatment in the animals, in the highland animals, well. And then there are other problems to follow this line of research. Rana temporaria population, overall population decreases everywhere in Europe, I think. And legislation regarding the experiments became stricter, so we don't continue to play with these animals. And my colleague is still active. She does things from time to time, but I may tell here on an illegal basis. I mean, without, well, yeah. But she shifted to plants, so plant experiments is a better thing. We collected publications and data, also raw data here in this website. And instead of catching animals, I prefer now walking in the highland areas. This is where I want to stop my presentation, where I hope you will clap your hands. And maybe somebody will tell if there is 1 or 2 questions time or not. Yeah.
Yes, of course.
No problem. I think we can hear you even without.
Thank you for a very nice presentation. I'm very happy that this conference takes place in Slovenia. And I'm Maruša Hribar from Slovenia. I would like to ask you regarding your presentation. We nicely saw application of Aaron Schulz law and hormesis, because you nicely presented that a high dilution block organ and low potencies, let's say 5CH, stimulate organ, and the middle frequencies, let's say 9CH thymulin, it's thyroid hormone, moderate immune system. Do you believe that we can apply this Aaron-Schulz law for all hormones, let's say for estrogen, for insulin, for adrenaline? Do you believe that all diluted low potency hormones can stimulate organs and the high dilutions can block organ? What is your impression?
Yeah, well, just from the non-scientific impression, I'd say yes. Yeah, that's the idea. From our experiments, I can say, and I see Pierre Dorfman smiling because he always asked me, what is the practical use of what you do? And I said, no practical use. Please don't bother me with practical use. It's just a theory. It's just, or you're just observing what happens if you do this and that. We're just playing. So, but the Aaron-Schulz law on hormesis is, of course, for us was basic, and as biologists, what the basics of what we did. And well, I think that also with a high potency, you can have an accelerating effect, like a drug proving. So I think it's not automatically that a high potency slows down, and so but the high potency is opposite, the effect is opposite of that of a low dilution. So you can have a drug proving effect if you apply too often, for example. And I don't know where the break is, where the molecular doses of thyroxine or any other hormone would be counterbalanced by the isopathic or homeopathic effect. So you have to take into account the molecular effect and then the isopathic effect. I don't know where it would switch, but maybe in the range of 20 or 25 or something.
May I say my opinion?
Hi.
I've Catherine Gaucher. I verified that with hormonal homeopathic remedies, not similitude, but hormonal, which is not the same. I verified that steroid, estrogens, well, mainly these two, I always verify this law, but not with similitude.
Mm-hmm.
I mean that if you put testosterone 30CH, you will low down the side effects of, well, the extra effects like acne, loss of hair. It is very useful because now all the allopathic remedies have too much side effects, so our colleagues don't know what to do. And when they send them to me, people get difference. Some symptoms disappear, even if biological doesn't change. Sometimes it does, sometimes not, but symptoms disappear, which is not always the same with with, for example, Ignacia or Lycopodium. I'm not sure, but with thyroid, with folliculinum, with testosterone, I always verify that. Thank you.
Thank you for this contribution. I think it answers well. Yeah.
Hey, Christian, thanks for that lovely talk. You mentioned the effects through glass, and then within the same breath, you said it's an effect of the experimenter. Could you expand on that and how you went through some effects seen through glass to the idea that there was an experimental effect? I'm curious.
If I was to expand on this one single breath, I would expand in another direction and use to quartz glass proved to be much better suited for the experiments than normal glass. So I think this is something that can be said. Quartz, quartz glass seem to be better suited for the transfer from the inside digoxin high dilution to the basin water. So yeah, quartz glass, which is quite expensive because this is why we didn't use it in the beginning. So this is one thing I would answer here. And the other thing is, and this is really a different question, different thing, that the experimenter, that we've started to feel that, we started to feel we should maybe make pre-tests with the experimenters, with these people who do the experiments. I would say, well, this is really as an answer to your question at the lunch break, yeah? I would say active people, I did jogging, running, maybe it makes life easier, overall life easier. Maybe it makes your influence on the homeopathic experiment easier when you have some energy, so to say, to put into the system. I don't know. And the other thing is Waltraud. She's a gardener by training, my colleague. She has the green thumb. She does with plants. Everything works. Everything grows. So this is also maybe a feature that is good for a homeopathic experimenter. This is a vast topic, and you would have to experiment on that, of course, with different people and different targets and different measurements, and to know how to assess that all before presenting this to anybody in the outside world. Yeah, because otherwise it's just self-immunization. It doesn't work with you because you don't believe in homeopathy. It works with me because I believe. So this is not what we want to. And I don't think it's wise to present homeopathy as a special area of parapsychological research. It's not really wise to do that, but maybe in 200 years, and this will be integrated into main science or whatever. Before I'm stopped now, I want to convey greetings of a dear friend, Menachem Oberbaum. He's GIRI member from, I don't know, from the first year, I think one of the founding members. Yeah, from the beginning. And we're in exchange. And he asked me to give his greetings here at the conference. And I want to, well, this is a perfect link to the hormesis topic, because he was the one who started us biologists with the Hermetic idea. He just, and his team, including scientists from India, published, I think it may be a crucial thing, homeopathy versus conventional primary care in children during the first 2 years of life, which was a nice study, in the European Journal of Pediatrics, which is a normal non-homeopathic study journal. So I think he deserves to be presented here also. Yeah. Okay.